Good afternoon, class. Welcome to Antimicrobial Drugs 2. So today I want to talk about the difference between prebiotics and probiotics. So prebiotics, so last time remember we talked about superinfection and how that can lead to C. diff and yeast infections and things like that. So one thing we can do to kind of protect our microbiota or our natural flora is through prebiotics and probiotics. So what's the difference? Prebiotics are nutrients that encourage the growth of beneficial microbes. And then probiotics are actually live cultures. So this would be like yogurt or lactobacillus inserts. And so that's the difference between prebiotics and probiotics. But they're very good. They're also supposed to be good for food allergies according to your book. So if you have those, that would be neat. Okay, so let's talk about the mechanisms of drug resistance. So we talked about how some drugs were so wonderful because they would target the cell wall. This would be things like bacitracin and ampicillin and the penicillins. But how do we get this drug resistance that came up almost immediately? So some organisms are naturally drug resistant, and so there's several ways they can become resistant. So we're going to use penicillin as an example in the first one. So one is a lot of organisms can make something called penicillinase. Penicillinase, remember it ends in A's, so it's an enzyme. It's a protein they make that essentially chews up penicillin. So what it does is it attacks this part called the beta-lactam ring, and then the penicillin doesn't work because it's chewed up. So that's called chew it up. Then the second way, so this is the cell wall of the bacteria. They have receptors that the drug will target, and then they'll take it in and the drug can destroy the bacteria. But if we change by a mutation the shape of the receptor, then the drug can't get in that way. So that would be called keep it out, and that's by mutation. And then if we go down here, we have another scenario. The drug does get in, but a lot of bacteria have pumps. They'll have a pump. As soon as it gets in, they'll pump it out. So this is called pump it out. And then another way is if the drug is targeting one of the steps in the bacteria's metabolism, they'll just change their metabolism by mutation. So if the drug works right here, they'll have an alternate B, C, and then they'll still make the same product, so it won't bother the microbe, the bacteria usually, or the protozoa, whatever it is. And so that's called do without. So they can chew it up, keep it out, pump it out, do without. So how do they do this? Usually it's through conjugation. So remember, they make a conjugation pilus, and they can take a resistant plasmid, also called an R plasmid, that has one of these abilities and transfer it from one bacteria to another. Also there's transduction. That's when bacteria is attacked by a bacteriophage, which is a virus, and if it picked up a little bit of the virus or a little bit of the antibiotic resistant genes from the last bacteria it had infected, it can bring it to the new bacteria. Another way is transformation, where bacteria have this ability to take up DNA from their environment. Like if a lot of bacteria have died, they can take their DNA in and use those. If the DNA are plasmids that have resistance, then so it's quite easy to do. Also, last video, I forgot to mention that a lot of people have an allergy to penicillin. So that's on top of antibiotic resistance. This is when your body actually treats the penicillin as an allergen. So kind of like poison ivy. So the first time you take the penicillin, your body becomes not resistant, but allergic to it, but it doesn't bother you that much, just like the first time you touch poison ivy. But then the T cells get involved, and the second time it can be bad, and you'll be covered with a rash. So that's why the doctor will ask you, are you allergic to anything? They're looking for penicillin specifically or other drugs. So I also forgot to mention when we were talking about the Kirby-Bauer test and the test where you take the antibiotic and you have increasing dilution, that is called MIC. So you're trying to find the minimum inhibitory concentration, MIC. And what that is, so you know the minimum amount of drug you can give the person that will still affect the bacteria. Why do they do that? Because a lot of bacteria that don't have as good of a Selective toxicity, like for example, bacitracin or ampicillin or the penicillins have very good selective toxicity. You want to give the patient the least amount because most antimicrobials are toxic to the patient, at least a little bit. So we have something called a therapeutic index, or TI. The therapeutic index is a number that says how toxic that antimicrobial is to the pathogen compared to the patient. So if you have a very low number, for example 1.2, that is very bad. It means it's almost as toxic to the patient as to the bacteria. If you have a nice high number like 10 or 20, that's better. That means it's not as toxic to the patient as to the pathogen. So that's called the therapeutic index, or TI, and so that is antimicrobial drugs.